All right, everyone. Dr. Phil here. Is like June 1st, Tuesday after Memorial Day. So I want to go over real quick. Let me. Stop sharing. All right, I guess it's working. All right, so I want to go over chapter 5 quick on eukaryotes. So remember, I said eukaryotes means true nucleus. So these are going to be different than the prokaryotes. And a lot of times with micro, we tend to think of the micro just being bacteria or something very small. But we are going to be dealing with eukaryotic organisms that are pathogenic, and they're going to be finding their way into us or on us, either as a mold or something we consume. It could be a flagellate in our blood, or it could be a malaria, or it could be a fungus growing in our skin, or ventilatum from ciliated from pigs. We'll get into that. I'll give you a little chart at the end from your book. Not that I'm going to care about it at this moment, but when we get into the stuff towards the end of the semester, you will at least have heard it before, so it won't be completely new. But just realize that the eukaryotes appeared approximately 2 billion years ago, and I said in, I think, chapter 1, if we were looking at a clock between midnight and midnight, we showed up. All right, humans, we showed up late to the party at 11:59 p.m., but the eukaryotes showed up about 2 billion years ago, but well after the prokaryotes. And then I mentioned that we have a strong suspicion that eukaryotes developed from prokaryotes. So a prokaryotic organism engulfed another prokaryotic organism, and that's how we have eukaryotes with the organelles. And our thought process is, remember I said that prokaryotes have a different ribosome, a 70S ribosome, as opposed to the eukaryotes. And the only exception to that was in eukaryotes, our mitochondria RNA is the same as mitochondria in a prokaryote. So that's kind of how we figured that one consumed the other. So evidence suggests evolution from prokaryotic organisms by symbiosis. Organelles originated from prokaryotic cells trapped inside of them. So each one of these prokaryotic organisms, they traded off symbiotically. They decided that one may be good at processing food, and one might be able to be good at processing energy. So they kind of evolved into the organelles that we have now. Okay, so this is new. So there's some ancient eukaryotic cells, and just realize that in comparison to bacteria, this is going to be relatively large. And when we start talking about the eukaryotic worms or some of the flagellates, they're going to be humongous. Like Giardia and some of these other organisms are going to be extremely large in comparison to the tinier bacillus or bacteria. Right. So I downloaded this in a new format, so it might be a little bit different. I don't have arrows now. I have to just swipe on the MacBook. All right. So eukaryotic organisms studied in microbiology. So protozoa, they're always unicellular. They're very, very large. We'll talk a little bit towards the end of the semester on molds and fungus. Remember that these are dimorphic depending on the temperature. And then the really, really large organisms like the Helminths, or worms, roundworms, pinworms, tapeworms, lumbricoides. These are going to be massive. You can see them with the naked eye. So I said last video I did to review the eukaryotic cell. I know we're talking about, there was a picture of a bacterium there, and I kind of said not every bacteria or not every eukaryotic cell is going to have all of these structures, but here is a sort of a 3D representation of a basic cell with all these different parts. Now realize if a cell is going to be breaking down lipids or if it's going to be producing proteins, it's going to need more ribosomes. Right? So whatever the organism is producing, what its main function is, they're going to have more of one organism. Like Golgi apparatus is going to be processing enzymes or peroxisomes or something. It would have a lot more Golgi apparatus. We'd have to package those and then excrete them through exocytosis through the cell wall. Right? And then if an organism has a lot, it's very, very active, it's going to have a lot more mitochondria. And I mentioned before that some of these organisms like the distal convoluted, distal, yeah, in the kidney. So those can produce mitochondria at will. Just anything that needs to have a lot of energy production, they generally can just make more mitochondria if they need to produce more energy. So in Blackboard, there is in Unit 1, there's a video, I think it's by Nucleus, on the parts of the cell. So just read through that or look through that. This should be review from your human bio or A&P or whatever it happens to be. So basically know what each cell part does. And if we ask you a question, you know, what like a liver cell is producing a lot of enzymes or proteins that would have more of which organelle. So there's a chart from your book, just kind of showing eukaryotic cells, and kind of a flow chart. So external organelles and other structures. In humans, the only cell we have that's flagellated is this sperm, but there's other eukaryotic cells that have flagella or cilia. And a lot of times they think it's for motility, but sometimes the cilia can help feed them if it has a wave-like motion. Cell wall, cell structures, things like that. Glycocollax, I said before, that's just sugar coating, capsules and slime layers to help you with dehydration and biofilms and adhesion to certain structures. All right. So external structures, flagella, we talked about that previously, or I talked about it in one of the videos, but just realize these are long sheath-like cylinders containing microtubules in a 9-2. So if you ever see it, they'll ask you the arrangement, it's going to be 9-2. So if you can remember, you know, I don't know if you guys are visual or audio or a little bit of both, but if you can just picture that, there's 2 there and then 9 around it. So that's the arrangement. Right. These are much, much thicker than prokaryotic flagella. All right, and these are going to help with motility. And these really can't do a run and tumble like the bacteria. They kind of whip like a snake. Gives them some motion. They really can't back up too much. Right. And then external structures, locomotion appendages, cilia. So they're going to be much, much shorter. And you know, these are for motion, and we'll learn about diphtheria and pertussis and some of these things and how they can affect the cilia of our cells. These are similar to overall structures of flagella, but shorter and more numerous. Found only on a single group of protozoa in certain animal cells. These function, like I said, is set in motility, feeding, and filtering, and I think it might be ventilatum. But it's going to feed it into this oral groove or gullet. Bacteria will swing like a sweep-like motion, like the wave, into the orifice for consumption. Glycocolax, remember I said before, glyco is always sugar, and the outermost boundary that comes in direct contact with the environment, usually composed of polysaccharides, appears as a network of fibers, a slime layer, or a capsule. So these can be for adhesion, like I said, they can be biofilm, they can be for eluting the immune system. All right, function and adherence, protection, signal reception, helps it from dehydrating. And then beneath that, fungi and most algae have a thick, rigid cell wall, so they're very, very structured. In lab, you probably looked at some of the plant structures, and you probably noticed that they were enormous and relatively very easy to see and structured because of their large size. Protozoa, a few algae, and all animal cells lack a cell wall and only have a membrane. And once again, that membrane is what determines what comes in and out of that organism. So if we can find a way of disrupting it, we can definitely hinder the reproduction or survival of that organism. Okay, Phil, now what have you done here? Swipe it. Well, there we go. Okay, cell wall, rigid, provides structure and support and shape. So that should be review for everyone who took human bio. Okay, have a thick inner layer of polysaccharide composed of chitin or cellulose and a thin layer of mixed glycans. We don't really care about that per se unless we had some kind of a fungal infection and we had to break it up using estradiol in the cell wall as opposed to the cholesterol that would be in our cells. So we have to find a way of destroying that cell wall, hopefully. And remember too that the problem with eukaryotic cells and us as hosts is we're eukaryotic cells too. So there's not that many discrete differences between eukaryotes and us. So the more similar we are, the harder they are to get rid of, because whatever is going to affect them is also going to affect the host cell. Algae varies in chemical composition. Substances commonly found include cellulose, pectin, mannans, silicon dioxide, calcium carbonate, relatively durable. And then the cell membrane. All right, so this is really critical for survival. And typically a bilayer of phospholipids and proteins, just like us. Sterols confer stability, just like in our cells, we have cholesterol. They have sterols or estradiol, and some of these other organisms serve as selectively permeable barrier. Transport eukaryotic cells also contain membrane-bound organelles that account for 68% of their volume. Very similar to us in more ways than bacteria. Nucleus, convex sphere, most prominent organelle of the eukaryotic cell. You should have been looking at these in bio. And the nuclear envelope composed of two parallel membranes separated by the perforation with pores, and that is how we can get the genetic information out of the nucleus without pulling out the actual DNA. Contains chromosomes. The nucleolus, they ask you about that structure, the dark area for RNA synthesis and ribosome assembly. Excuse me. So if you see that, you realize that that organism is probably going to go under some type of genetic coding or copying at some point if you see that all bound up like that. Nuclear changes during mitosis. All right, so these should be completely reviewed for everyone. You know, they line up, they separate, and then you should have two distinctly complete organisms. It's very, very similar. Two daughter cells. If there's a problem here, then we can say that, you know, some kind of genetic alteration, and if subsequent alterations keep happening, then we're going to open ourselves up to metaplasia or some other kind of thing. So once again, I said we're going to be going over the internal structures, and this should be a review. Watch the video, but they'll ask you, or I will definitely be asking you a question or two about this. how, if you know what the internal structures are, I'll ask you some version of that. So rough ER, rough endoplasmic reticulum, originates from the outer membrane. So this is continuous with the outer nuclear envelope and continuous network through the cytoplasm. Remember, rough ER, whenever you see rough ER or ribosome, you got to think protein. Protein synthesized and shunted into the endoplasmic reticulum for packaging and transport. First step in the secretory pathway. So if we had an organism that was secreting an enzyme, enzymes are made out of proteins, they'd have to be able to secrete it. We have to shunt it, we have to move it, so now we have to get a Golgi apparatus involved, vesicles, we have to get to the cell wall, and through exocytosis, release it into the interstitial fluid or the external environment from their perspective. Right? Smooth endoplasmic reticulum is closed tubular network without ribosomes, so it's not going to be, main function is not going to be protein. Functions in nutrient processing, synthesis, and storage of lipids. So whenever I think of smooth ER, I'm always thinking of hepatocytes. It's dealing with like, what would be similar to a liver function. So it's processing nutrients, synthesizing things, storing lipids, not making proteins though. Right. There's the polyribosomes, poly meaning many. There's a nuclear envelope. These are continuous. And we're going to bring this up again. As we're running this messenger RNA through the ribosomes, it's going to read into the codons, and they're going to be attaching these amino acids in a certain sequence, providing the proteins. That's how it's going to work. So we're going to spend a little time with genetics and figure out how, if you're a bacterium or eukaryotic cell, can use tetracycline or some of these other things that disrupt the ribosome attachment of the amino acids. Golgi apparatus modifies, stores, and packages proteins. It's kind of like the Amazon kind of ships them out. It consists of a stack of flattened sacks called cisternae or cistern. Back in the day, they used to use those to collect rainwater. Some more internal structures. We have transitional vesicles from the ER, endoplasmic reticulum containing proteins, go to the Golgi apparatus for modification and maturation. And I said we need some kind of vesicle to transport it. Condensing vesicles transport proteins to organelles or secrete proteins to the outside. So nucleus, rough endoplasmic reticulum, protein. Golgi apparatus is processing, packaging, wrapping up nice and pretty. Vesicles are going to carry it to the cell wall, and then through exocytosis, we're going to be secreting that into the external environment. Lysomes. So when you think of lysomes, these are going to be proteins. So these are going to be digestive proteins, usually. Vesicles containing enzymes that originate from the Golgi apparatus, involved in intracellular digestion of food particles, and in protecting against invading microbes. So these are going to be digestive enzymes. So we could engulf a bacterium, macrophage or a monocyte could engulf it, and then if we hit it with digestive enzymes, rather than digesting food for the cell, or we could too, it would digest that bacterium and will reclaim any part of it that's usable and will excrete the rest. So vacuoles are membrane-bound sacs containing particles to be digested, excreted, or stored. So we're going to trap these organisms, if we're in the immune system, in these vesicles, and we're going to hit it with peroxidase or some of these digestive enzymes to destroy it. Phagosomes means to eat. Vesicles emerge with a lysome, and then we digest it and consume it. Mitochondria, we'll be talking about that. I'm sure you all learned this powerhouse of the cell, ATP production. And then, if we're talking about eukaryotic cells, it happens in the outer membrane. If it's happening in bacteria, it's going to happen in the cell membrane because it doesn't have an outer membrane. Function and energy production consists of an outer membrane and an inner membrane, folds called cristae for surface area. And just realize that you're going to be running the proton motive pump through here with hydrogen producing ATP and carbon dioxide. We'll see that in chapter 7, I think. All right. Because they hold the enzymes and electron carriers for aerobic respiration. It's going to run those positive hydrogen charges, running it through the membrane, and then back down through. It's going to be like a turbine. It's going to turn around. It's going to turn ADP into ATP, continuously running it. So after we phosphorylate the ATP to ADP, we run it through the proton motive pump, and then we turn that ADP to ATP. But we'll see that coming up. Divide independently of cell, all right? So the cell could divide when it needs to. Mitochondria can also divide if they need to ramp up energy production. All right. Contain DNA and prokaryotic ribosomes. Okay, so remember, these contain DNA and prokaryotic ribosomes. So this is a eukaryotic cell, but in the mitochondria, it has a prokaryotic ribosome. Very, very key function, because whenever we're hitting these microbes with anything that alters the ribosome, affects the ribosome and protein synthesis and production, we can start affecting the mitochondria in our own cells. Chloroplasts, that's basically plants. I don't like to spend a lot of time on those. Short of, we can be talking about oral thrush candida or some of these tinea pedis, or some of these things that are growing, transient things that are growing on your skin, or candida albicans can grow on your tongue for oral thrush or yeast infection, but usually people have to be immune compromised or something else going on. It wouldn't be an opportunistic organism. It generally wouldn't affect the human, because we have enough other stuff going on keeping it at bay. I'm not saying it's not in the body, but we have other things going on. It doesn't have a chance to overgrow. Chloroplasts convert the energy of the sunlight into chemical energy through photosynthesis. And please realize, when we talk about the Krebs cycle, please realize that all energy in the world, our world anyway, comes from the sun. So whether it's sun producing plants, and then the animals consume the plants for energy and nutrition, and then we consume the animal. The actual source is the sun, and we can run that whole Krebs cycle back and forth. All right. Either way, it's found in algae and plant cells. Outer membrane cover into membrane fold stacks called thylakoids, stacked into grainae. So it kind of looks like, a bunch of watch batteries or whatever you want. So they're called thylakoids, and they're stacked into grain, like grain elevator. Here. Primary producers of organic nutrients and other organisms. All right. So ribosomes, I'm going to spend some time on that with genetics. Relatively important structures as far as I'm concerned. Composed of RNA, and proteins scattered in the cytoplasm and associated with a rough ER. All right, so when we learn about genetics, please realize that these amino acids are floating around in the cytoplasm, whether it's bacterium or eukaryote, all right? And these ribosomes, we always show them together, but they're separate. They only come together when they're needed. All right, so the small one comes up, the big one docks with it, messenger RNA runs through, we start running the codons. These are larger than prokaryotic ribosomes, 80S as opposed to 70S, and these function in protein synthesis. Cytoskeleton, so these are basically there just for structure. Some of these organisms, these things on the larger organisms, they can zip line on these. So, the shortest distance between two places is a straight line. So they could theoretically zip line, but this is really there for structure for them. Flexible framework of proteins, microfilaments, and microtubules form network throughout the cytoplasm, involved in movement of cytoplasm, amoeboid motion, transport, and structural support. Right, so here's just a comparison from your book. This is from Tadaro, comparing prokaryotes, eukaryotes, and viruses. So we can talk about size, shape, structure. Okay, but viruses are smaller than prokaryotes. Prokaryotes are smaller than eukaryotes. So, nucleic acid are in all of them. Viruses really are just nucleic acid and some kind of a membrane or an envelope. All right. So, you could say, some people will say they're alive, some people will say they're dead. And that's really more for chapter 6 of the book. And then, how do they go through reproduction? Right. So is it mitosis? Well, that's only in eukaryotic cells. Prokaryotic cells, they use binary fission. Viruses have to hijack something else to use the machinery. And there's just some of these characteristics and where do they belong? Right. Okay. So just super quick survey of eukaryotic microbes. This is not by any means the information for the course, but just where would these organisms pop into um, discussion here? So fungus, fungi, algae, protozoan, and parasitic worms. Like I said, I'm not really concerned about algae. It's not going to find us hospitable. Fungus, maybe some molds and fungus, whether we're talking about something growing on the skin or if we're breathing in spores, coccidioidomycosis or something where we're kicking up some of these spores, we're breathing in the lungs and they're forming spores or some will have like a wagon wheel type shape, or they're forming large particles in the lungs. But generally, once again, this is somebody who likely was immune compromised or they got a very, very large infective dose. Right. So kingdom of fungus, there's 100,000 species divided into 2 groups. All right, so macroscopic large would be mushrooms, puffballs, gill fungi. Microscopic, what we'll be dealing with here would be your molds and your yeast. Majority are unicellular or colonial. A few have cellular specialization. When you come in, I ordered some tubes of penicillium. We'll see that. So when you bring your plates home, we'll leave them out to the environment. So find some place that's damp, maybe a bathroom or a basement or someplace where there's a little bit of extra moisture. Leave your plates out. Hopefully some of these spores will drop and we can grow them. We can compare them against some known, and we can just kind of guess what it might be. So once again, fungus is dimorphic, mold in the cold, yeast in the beast, depending on the temperature. And these fungus, if they get into your body, they don't want to go deep in the tissues. It's too warm for them. They're going to be really, really superficial. Even Sporotrixis from rose thorns might live in the lymph nodes a little bit, but it doesn't want to go deep. So yeast, your round ovoid shape, asexual reproduction. They have hyphae. And if you've ever seen bread mold or whatever, you've seen these. Long filamentous fungus or molds. Some exist in either form. So they're dimorphic, characteristics of some pathogenic molds. Right. These are all heterotrophic, so they are consuming dead something. So if it's mold growing on your shoes in your basement because they're living off the shoe leather and it's damp, or mold going on bread or other surfaces. Majority are harmless saprophobes living off dead plants and animals, and we kind of want them in the environment. We want them to break down things, even if it's breaking down stone and rocks in the dirt. Could be bioremediating some kind of garbage. Some are parasites living on the tissue of other organisms, but none are obligate. And if you have a fungal infection, it is now going to be called a mycosis. All right, so if you're Like if you have food poisoning from Staph aureus releasing toxins, I said before, that's a food intoxication. It is not a Staph aureus infection. All right, so you're getting sick from the secretions. You're not getting sick from the organism multiplying in your bloodstream or your system. Right? These are extremely widespread distribution in many habitats, and could be something as, I don't want to say innocuous, or as common as jock itch, athlete's foot. So depending on the area where it's finding the moisture, all right, it could be the same organism. It's just really contingent upon where's it growing. So yeast are soft, uniform texture and appearance, reproduce through asexual process called budding. So this is kind of similar to binary fission, but if you look, there's a marked difference in size. All right. We do this with yeast, growing yeast, yeast for bread or yeast for fermentation of alcoholic beverages. Fungal organization, there's filamentous fungus, massive hyphae called mycelium. It's a cottony, hairy and velvety texture. And you'll see this in lab, or you could look up Google images or on bread mold or whatever. It's going to have that real velvety, it's called the mycelium. Hypha may be divided by cross walls, septum or septate. That should be common. We have a septum, nasal septum, you have atrial or ventricle septum in the heart. So it's really going to be dividing wall. That should be pretty standard. Septum separates these. Vegetative hyphae digest and absorb nutrients. Vegetative, whenever we're talking about a vegetative bacteria or vegetative cell, generally it means it's just a normal, I don't say normal, it is a cell or bacterium just living out. It's like it's not literally going into trophocytes, a feeding phase. It's not going into the reproductive phase. Now, reproductive hyphae produce spores for replication. And, this is what used to freak me out when I would be at a restaurant or whatever, and I would see these ceiling fans or whatever going, there'd be dust and stuff all over them, they're blowing, and I'm thinking, well, this fan is blowing spores all over my food. So fungal reproduction, primarily through spores formed on reproductive hyphae, or asexual, right? So sometimes they can go through two different species can kind of join and they change the genetics a little bit, or can be asexual. Spores are formed through budding or mitosis, Candida or sporangiospores, and we'll see that coming up. So here's some types of asexual mold spores. All right. So these are going to just burst, sending spores all over. All right, so fungal reproduction, sexual reproduction, spores are formed following fusion of two different strains and formation of a sexual structure. Zygospores, ascospores, basidiospores. All right. Sexual spores and spore-forming structures are one basis for classification. Like I said, we're not going to spend a lot of time on that. All right, and there's some basic bread mold. You can see how there's asexual, right, or there can be sexual depending if another species mixed. This would be like mushrooms doing that. And there's some classifications of it. I said I'm particularly not that concerned with all of that per se. There's just some diversity of fungus. So, some of this is edible, right? And please realize we need this to produce, well, we should use it for penicillin, we need it for food, we use it on biogenetics, because these are large, and yeast cells are huge, right? So they can, if we can hijack them, they can produce all kinds of things for us. Macroscopic and microscopic observation. So when you bring your plates in, we'll be looking at spore-forming structures with a magnifying glass. Get out the dissecting scopes if you want. The hypha types, or what does that hypha look like? colony texture and pigmentation, just like when we look at our colonial morphology. Hopefully today and Thursday in lab, you brought your plates home. So we'll kind of be looking at colonial morphology. You know, is it raised? What color is it? What's the sheen on it? What does the border look like? Physiological characteristics and genetic makeup. We're not going to get into genetics, but we can just get an idea of what these are. When you bring your plates in, you know, we're not going to be genetically testing them, but you can look and see what are you guessing it could be. Right? So the roles of fungus, the adverse impact. So, you know, how could it potentially be bad for us? Well, mycosis is a fungal infection, allergies, toxic or toxin producing. So some of these are poisonous. Destruction of crops and food stores. So a lot of times people say, you know, there's a food shortage or they won't eat radiated food. But please realize, especially you know, like now, well, and it doesn't matter where they come from, but fruits, sometimes vegetables are coming in, you buy them, they look fantastic, you rinse them off, eat a few, put them in the refrigerator, pull out that half quarter or later, and there's mold growing all over it. Right. So short of, you know, doing a real super dilute bleach bath before you put them away, mold can destroy a lot of things. Can destroy things, well, like tomato sauce in your refrigerator. But the chances are, by the time mold's growing on it, that's been in there for 3 or 4 months. Time to get rid of it. Now, the beneficial impact, decomposes of dead plants and animals. We want those for bioremediation. All right, but the cool thing is, was a source of our antibiotics initially. We used to use it for alcohol, organic acids like acetone and things like that, vitamin production, and then used in making foods and in genetic studies. So I said before, a lot of these can be similar. It really depends on what their location. So derm, like dermatology, is skin. Dermatophytosis, so this would be something growing on your skin. Epidermis, hair, dermis can be attacked. Ringworm, the body, feet, athlete's foot, toenails. Same kind of organism, just we name it depending on its location generally. Mucous membrane, skin and nail, so candidiasis, so oral thrush or yeast infection, oral thrush of the mouth, or yeast infection. Females, same organism. Candida albicans, albicans meaning white. And then systemic, so deep. And I said that these don't like to go real deep, so you're going to breathe them in, or they're going to be in your skin or very, very superficial. Coccidioidomycosis, San Juan Key Valley. So these are going to be where you're dirt biking, you're digging up stuff, and you're breathing in these spores, and they're germinating in your lungs. Coccidio, yeah, imitans, North American blastomycosis. Right? That's in North America. And then histoplasmosis, that's breathing it in, the spores, that's in the Ohio Valley. That's really contingent on the temperature, right? So it means how severe are the winters, you know, what are the spring like? It's very, very temperature dependent. Cryptococcus, all right, and then lung or skin. Paracoccidiomycosis. So very, very similar, para meaning sort of, kind of, maybe, almost, but this is South American. Right? And this is what when I went to Peru, I was afraid I was going to get something like this. And when the storm kicked up, it's crazy. And there's some talk about some algae and protozoan. Algae is not really going to be affecting us at all. Protozoan, yeah, unicellular eukaryotes that lack tissues and share similarities in cell structure, nutrition, life cycle, and biochemistry. And they're eukaryotes, so it's really hard to get rid of them. So like Giardia, some of these other large protozoan that are eukaryotic can be a little bit more difficult to get rid of. There's your algae. Right. Most are free living in fresh and marine water. Plankton, they're usually a food source. Right. Provide basis for food web in most aquatic habitats, produce large portion of atmospheric oxygen. And I mentioned the other day that, you know, we all learned that trees and plants produce, you know, all the oxygen. That's true, but the majority of it's usually produced by algae, plankton. dinoflagellates can cause red tides and give off toxins that cause food poisoning with neurological symptoms. And the issue with that is when this happens, a lot of people, and I don't know if it still happens. A student maybe 5 or 6 years ago, we were talking about this, and I honestly can't remember. It was a South Asian country, and she said that they had the red tide come in and the fish were consuming it, and they kept telling these people, do not eat the fish, you'll get sick. And they didn't believe them because they had been eating this fish for generations. Like, who are you to tell me I can't eat that? But a lot of people got very, very sick or got a lot of neurological symptoms from eating fish that had consumed this. There's some algae classifications. Like I said, I won't be testing you guys on that at all. Not important in my book. All right, protozoan though. All right, so these are going to be very, very large, unicellular. They move on. Colonies are very rare. They're independent. They're moving around. Most are harmless, free living in the moist habitat. Some are animal parasites, can be spread by insect vectors. So remember, vector is something that flies, and vector comes from straight lines. So it's picking up one thing, flying somewhere, infecting another. All are heterotrophic. All right, they lack chloroplast. It means they can't produce energy from the sun. So they have to live off of something else. All right. Feed by engulfing other microbes and organic matter. And there's your protozoan. So these are going to have, I said, vegetative. Trophocyte is always the eating phase. Motile feeding phase. Most have locomotion, flagella, cilia, or pseudopods if it's an amoeba. So they have some way of moving, and they're going to really try to move towards a food source or move towards something that they find beneficial. Many can enter into a dormant resting stage when conditions are unfavorable for growth and feeding. This is the cyst form. So whenever they say trophocyte or vegetative, that is a feeding phase. When things get lean or things start happening, they insist or they make a cyst, and they're hoping that they'll be excreted or picked up by something else with a new environment and have a way of coming back. All reproduce asexually, like mitosis or multiple fission. Can also reproduce sexually, conjugation, like we saw with similar with the word that we were talking about, conjugation with the gram-negative bacilli, but that wasn't really reproduced. That was just sending genetic information for valence factors. And there's a breakdown of the protozoan identification. Classification is difficult because of the diversity, and this is really just, we had to think of a way. How do we group these? What's our, we tend to be able to memorize things better if we can at least group them into something that makes sense rather than just randomly memorizing a bunch of stuff. So Mastigophora, primary flagellar motility, some flagella and amoeboid action. Sarcodinia, Ciliophoria, there's obviously going to have cilia. Apoplexa, we'll see a bunch of these. His motility is absent except male gametes through locomotion, sexual and asexual reproduction, complex life cycle. So all are parasitic, and we'll talk about those later. So there's Mastigophora. So we're gonna identify this primary flagellar motility, some flagella and amoeboid sexual reproduction. So there's our friend Giardia. All right, those are not eyes; those are nuclei. Right? Nasty. Sarcodinia, Spiphoria, they're gonna have cilia on them. All right, Apoplexa. All right, it's gonna be the food vacuoles. And sometimes they kind of move the food or whatever towards their gullet or gullet, I think they call it. All right, so these are the ones we're going to be talking about later on. Entamoeba histolytica, worms, protozoan, really. Naegleria or Anthraconamoeba. So this is all going to be infection from swimming. So whether it gets to the brain or not, we can differentiate these two. Endodosis, coli. These are zoonotic in pigs. So they'll say, what is the only ciliated protozoa to affect the human host? Right, so this is it right there. And they're going to say, you can get it from horses too, I guess, but it lives in the microbiome of a pig, and you'd have to get it from pig feces. So it'd have to be a pig farmer, or somehow they would have gotten, you'd have to be ingesting part of the pig's microbiome somehow. Giardiasis, all right, Trichomonas, vaginalis, talk about that. Hemoflagellates, so hemo meaning blood. So these are trypanosomes, so African sleeping sickness, things like that, where the red-footed bug is going to bite you. Leishmania, malaria, right, Plasmodium, vector borne through mosquito, female Anopheles mosquito, Toxoplasma gondii, Cryptosporidium. All right, this is from Free Living Water, and there was a big case in Milwaukee in 1990, 1991, 92, or whatever. Pretty rare. They don't know where it actually came from, but it did infect 100,000 people died from that. Right. And there's the trypanosomes, Brucei or Creutzei, so African sleeping sickness, Shigawa's bug. So these are going to be vector-borne. The bug is going to bite you. It's going to release these into your bloodstream. They're going to be swimming around your bloodstream. Right? So these can cause major brain damage, tumors, things like that. And to amoeba histolytica, amoebic dysentery worldwide is going to cause some major diarrheal issues. Right? It has to do with the digestive lining, secretory diarrhea. And then we'll talk towards the end, some parasitic worms, and these are going to be very large, parasitize their host tissues. They have mouth parts they can grip, and we have to find a way of paralyzing them or getting them to release. But their job is just to release eggs or release segments, hoping that someone else will pick them up and be able to perpetuate the species. And then we'll break them down into cestodes with the tapeworms, nematodes are the flukes. They're going to be much flatter looking, right? And the roundworms are nematodes. Right? We'll talk about tapeworms, the pork and beef tapeworm, how we're going to be dropping off these segments. There's the scolex, which attaches. They're dropping off segments. So each one of these segments, theoretically, if you picked it up and consumed it, you would have a tapeworm. Right? And if we were in class, it would tell you if you're going to get a tapeworm, you want to get the beef tapeworm because it doesn't leave your digestive tract. So if you want to lose a lot of weight quickly, just find one of these little segments. All right. Distribution and importance of parasitic worms. They're not really not important, but please realize that a lot of these other countries, these people can have more than one pathogen. They can have more than one thing going on through their whole life. It might be common in other countries, some parts of rural China and India, very common. And please realize they have a lot of diseases. They're reinfecting each other through feces or not boiling the water. So they just keep reinfecting each other. Acquired through ingestion of larvae or eggs and food from soil or water. Some are carried by insect vectors, affects billions of humans. Right. There's a life cycle of pinworm. We'll talk about that, how it's leaving the anus, leaving eggs, and then the child scratches. It's under their fingernails. They can touch food in the refrigerator or open the cereal box Saturday morning and infect the next participant of that box of cereal. Gross. And I think that might be it. So I'm going to close this down. I'm going to Process this.